Hi, welcome back to educator.com. This is the lesson on blood. Blood has numerous functions, not just circulating fluids. It's the transport of nutrients, sugars, proteins, lipids, vitamins, etc. Gases, oxygen and CO2 are among them, yes. Wastes, the ones that you end up excreting either through urine, through sweat, etc. And hormones, the way that hormones actually get to organs and cause them to change something about their physiology is through blood. Regulation of pH. pH has to do with acids versus bases. You have a zone in which your pH in your body needs to be maintained there. The approximate pH for the human bloodstream is 7.4, ever so slightly basic. If that goes down to like below 7, that's enough to kill a person because the pH scale is logarithmic and you want to keep it in a very particular range. So your bloodstream has a lot to do with regulating that. Restriction of fluid loss during injury. If you do have a cut, you're bleeding, you're hemorrhaging, some of the characteristics of your blood enable you to stop that. Defense against pathogens and toxins. White blood cells are the parts of the blood that are going to be primarily protecting you and killing off those foreign invaders. And regulation of body temperature. That's very important as well. And before I move on, I just wanted to show you what these are. This is your average red blood cell. This is a platelet, and this is a white blood cell. Now, more often than not, white blood cells or leukocytes are going to be much larger than red blood cells, but you do have a heck of a lot more red blood cells. The ratio of red blood cells to white blood cells in the blood is approximately 1,000 to 1. That's why your blood is usually red, because of the red blood cells. This is a micrograph. This is an actual image. It's not computer generated. If you were to take a blood sample from a person and centrifuge it, spin it around really fast, you would get a separation of all the blood parts by density. The densest parts, specifically what's called hematocrit. Hematocrit is all of these solid blood parts, majority of which is red blood cells. You're going to get that sinking towards the bottom because those solid particles and cells are much more dense. And of course, the fluid portion of blood is going to stay on the top. So when we look at all the portions, let's start at the bottom. Erythrocytes. Erythrocytes is the technical term for red blood cells. Erythro means red, cyte means cell. So approximately 45% of the average person's blood is the red blood cells. The other part up next is the buffy coat. There's this white band that tends to be a coming together of all of your white blood cells and platelets, which are just slightly less dense than your compacted red blood cells. So thrombocytes is the technical term for platelets, and leukocytes is the technical term for white blood cells. White cells. And we'll talk more about the functions of these later. Plasma. Yellow fluid. The particles dissolved in plasma is what gives it that yellow look. There are lots of particles dissolved in the plasma, the fluid, the actual fluid of your blood. Of course, a lot of plasma, most of it, is water. You also have gases. Oxygen, CO2, nitrogen gas, there's a lot of them. You're going to have nutrients. All those molecules that are meant to nourish your body. Of course, on the flip side of that are wastes, those waste products that are going to be eventually excreted. And then on top of all of that, you're going to have hormones. That's how hormones get to all the different cells via the fluid of your blood. And you also have dissolved blood proteins. Now, included in the nutrients are going to be amino acids, the proteins that you're feeding your cells with. Your cells need proteins to function and stay alive. These proteins, they stick around in the blood, and they all have their own purposes. The majority of these blood proteins, 60% of them approximately, is the albumins. Albumins are hanging around in your blood to help maintain osmotic pressure. If you took biology, osmosis has to do with the passive, or means movement without energy, the passive natural movement of water to where there's less water by concentration. If you didn't have as many albumins in your bloodstream as you do, there would be too much water flow. Think about this. Having blood proteins in your blood helps keep water there. If there were a lot less of them, you actually would sometimes lose too much water out of your bloodstream. So having albumins just helps maintain that osmotic pressure in your body. Next up, globulins. There are two globulins. There are two main kinds of globulins. The not most important one, but the one that you've probably heard most about is antibodies. So antibodies, it's a type of globulin protein. Those are very specifically shaped molecules that are meant to attach and help your white blood cells attack foreign invaders. So they'll attach to bacteria, viruses, to immobilize them and to mark them so that they can get destroyed, so that they don't harm you. So antibodies is one type of globulin. Another type of globulin is transport proteins. There are certain substances in your bloodstream that aren't necessarily getting along with water. They might be non-polar particles that don't dissolve well in the plasma. So globulins that are the transport variety will sometimes be like a little shuttler or a little raft getting them through the bloodstream. Certain hormones need to be attached to globulins. And finally, the next major kind of blood protein is fibrinogen. Fibrinogen is what you make fibrin out of, and without fibrinogen, you wouldn't be able to make a blood clot. And a blood clot is the way that you stop bleeding, and that's very important. So there's the components of plasma and the other blood components. So how do blood cells get formed in your body? Where do they originate? Well, hemocytoblasts tend to be hanging around in your bone marrow. And hemocytoblasts, that's a fancy term for blood cell maker. There's one major kind of stem cell known as a hemocytoblast that through mitotic divisions, by dividing, makes different lines or sort of lineages of cells. So for instance, where you get this from is the hemocytoblast. The proerythroblast eventually leads to the formation of red blood cells, erythrocytes. This particular lineage leads to a certain kind of a white blood cell of the leukocytes called granulocytes, because they have little granules in the cells. It looks like they have little dots, little particles in these cells. On the other hand, you have this other kind of white blood cell category called agranulocytes, meaning not having those granules. Monocytes and lymphocytes, they come from a slightly different daughter cell from hemocytoblasts called lymphoblasts and monoblasts. And finally, megakaryoplasts turn into megakaryocytes, and eventually the split up portions of that end up being thrombocytes, which are platelets. And all of these originate in bone marrow and end up in your bloodstream every second of every day of your life. Red blood cells, let's start with those first. They make up the majority of the hematocrit of blood. Erythrocytes, the technical term for red blood cells, are shaped like biconcave discs. So here's an image of what red blood cells look like up close. From a superior view of a red blood cell, they should look round, and there's a little indentation in the middle. It almost looks like a donut that doesn't have a full hole in it. If you were to take this and then cut it there, and flip the cells so you could see it on the side, they would look like this. Biconcave disc. You see that like both sides are concave, and it has this shape. Making up the majority of the protein inside of it is something called hemoglobin. And the interesting thing about the reason why the shape is this way, some of it has to do with the fact that they're enucleated. This is one of those rare examples in the body of cells that don't have a nucleus. So an interesting thing is that in addition to them not having a nucleus, they excrete it, they get rid of it before they end up flowing around in your bloodstream. They also don't have mitochondria, and they don't have ribosomes. So they're not doing a lot of the cellular activity that your cells tend to do just to stay alive. You know, these only last for so long. Eventually they get damaged, but the other reason why not having something like mitochondria is important in here is if these had mitochondria in them, and the purpose of red blood cells is to shuttle or carry oxygen through your body, if there was mitochondria in here, the mitochondria of the red blood cell could kind of steal the oxygen off of this little oxygen raft, and that would not be good. You want to have these things just delivering oxygen to the cells that are keeping your tissues alive. So they don't have a nucleus, no mitochondria. They're just jam-packed with this protein called hemoglobin that has a high affinity for oxygen. It's like a little magnet, in a sense, for oxygen. And this protein allows it to be like an oxygen raft. I like to think of oxygen just kind of sitting back and just enjoying its ride on the red blood cell until it gets to a capillary and ends up finding its temporary home. Oxyhemoglobin versus deoxyhemoglobin. Depending on whether or not oxygen is attached to the red blood cell and physically attached to hemoglobin, it has a slightly different color. When oxygen is on hemoglobin, it looks red. When there is no oxygen on hemoglobin, it tends to look like a deep, dark purplish that can oftentimes look blue. And that's how you get that look with veins, being slightly bluish in the systemic circuit, and arteries and arterioles being more reddish because of the particular part of the blood vessels that have oxygen attachment versus no oxygen attachment. And red blood cells last up to 4 months. Some of them last a lot less time than that because they can get damaged as they're moving through, getting shuttled through your bloodstream. They get damaged, they get ruined, and they break down. And since they don't have a nucleus, and since they don't have those other organelles, they're not repairing themselves. You're getting rid of at least 1 million red blood cells every second of every day. And you're also making that many. So you can get away with having these damaged red blood cells because you're making 1 to 3 million new red blood cells. And they have those entering the bloodstream every second. And this is variable because I've seen textbooks say 1 million. I've seen textbooks say 3 million. So there's a range. I'm sure it depends on the person, depends on their size, depends how many liters of blood are in their body. Breakdown and renewal of red blood cells. So in the liver, spleen, or bone marrow, that's where red blood cells eventually end their journey. They're engulfed by cells there, and then they, something called hemolysis, they hemolyze, causes them to rupture. They are broken down, and they end up, you take the hemoglobin molecule, which is right here. This is a kind of like an organic chemistry style model of the molecule. At each corner here, you have carbons. You can see where there are not carbons at the corners between these bonds: nitrogen, oxygen, hydrogen. In the center here is iron. Iron's a very critical element with the formation of hemoglobin. Well, what happens is, as the red blood cells are being stripped apart, the heme part of this hemoglobin, which is what the iron is needed for, that gets broken apart and ends up producing a molecule called biliverdin. Biliverdin looks slightly greenish, and so if you get certain bruises, you may have seen like some bruises have like kind of a greenish tone. That's because the pressure that caused the breaking of the capillaries was so great that you damaged and broke apart so many hemoglobin molecules in the red blood cells that the biliverdin ended up kind of leaking out into that interstitial space and giving it that green color. Eventually all that goes away because you have white blood cells that go up and swallow up all that excess stuff and clean it up. Eventually biliverdin is converted to bilirubin, and bilirubin has more of a yellowish, yellow-orange kind of tone. Bilirubin is related to jaundice. What ends up happening is bilirubin is supposed to be brought to the liver in your bloodstream, and then bilirubin eventually ends up in bile, and bile ends up in your digestive tract. But if your liver is not doing a very good job of dealing with that bilirubin, maybe blood flow to the liver or something's wrong with it, you'll get a buildup of bilirubin around the body, and it can end up showing through in your skin. And so jaundice is that sort of yellow look all over the skin and even in the whites of the eyes. Something like hepatitis, which is an infection of the liver, is going to lead to that happening, and that's because of bilirubin. Erythropoiesis makes new red blood cells, and EPO stands for erythropoietin. So erythropoietin is the hormone. Erythropoiesis is the process of making red blood cells. So that's going to be that one lineage I showed you on the previous slide that comes from hemocytoblast but ends up making red blood cells specifically. All right, when it comes to blood transfusions, you have to make sure that if someone needs blood, they've lost blood, you need to give them blood from a donor, there has to be compatibility. If not, you're going to get some problems happening, and I'll tell you how those problems occur. So let's look at the surface of each of these different kinds of red blood cells and these different blood types and what's also in the plasma and see how they go together. So if we looked around the surface of a red blood cell in somebody who has type A blood, let's say that on the surface there are these little molecules, these little antigens is what they're called, projecting through the plasma membrane. So this is an A antigen. Antigen is a general term for some kind of molecule. Usually it's proteinaceous, it's made from protein. Sometimes there are lipidy characteristics to them. But even the ones on the surface of a bacterium, a bacterial cell, you can call those antigens and use that term in the case of how antibodies that your body produces are going to be attached to there. Well, there's antibodies with respect to this too. So this is a red blood cell that belongs in this person's body, and they have this kind of antigen on the surface, and I'm giving it that kind of squarish shape. Well, the person with A blood has an antibody that goes against B blood. So right next door to it, you're going to have antibodies that look like this. Now, in later lessons in the immune system or lymphatic system lesson, you'll see that the actual structure of antibodies is more Y-shaped, but for the case of keeping this simple, I'm keeping it like this. So these two here are called anti-B, because they're against B blood, antibodies. And you'll see what this means in a sec, because now I'm going to show you what's on the surface of B blood cells. By the way, there would be a lot more antigens than what I'm drawing, but I can't draw that in this particular instance. I'm just keeping it simple. So this is actually a B antigen. So a B antigen, different shape, different kind of conformation, different molecular shape than this particular antigen here. Well, B blood in the plasma, in that fluid floating around, you have the opposite going on. You have antibodies that would look like this. I know this looks like an H, but use your imagination. So these would be floating around, and these particular antibodies, guess what? Anti-A antibodies. So the problem with somebody who has A blood giving them B blood from that other person is that in the person with A blood, they've got this floating around. They've got these and these. When you put this blood into their body, great. Now you've added red blood cells. That's what they were missing. That's what they need to stay alive. But you've also introduced anti-A antibodies. These are going to attach to there. They have a perfect fit, and it's going to cause red blood cells to clump together, because when antibodies see this little hole, this little space, attaches that antigen, imagine them just having antibodies all around them and then having other red blood cells clump together. That's called agglutination, and that's going to form an abnormal clot that doesn't belong there. The other problem is when you've added in this kind of red blood cell, look what they have in their bloodstream here. They've got the anti-B antibody, so that's going to cause a problem. And some people say, well, why don't you just take out the antibodies? But like I just said, just the fact that you're putting this particular cell into their bloodstream, you can't get rid of all the anti-B antibodies in that person's blood flow. It's just, that's not possible. So these don't go together. Next up, AB blood. The reason why it's called AB blood, it has both A and B antigens. It's got A antigens, it's got B antigens. But guess what? No antibodies to the A or B antigens. Now, there are antibodies in this person's bloodstream, yes, but they're not going to naturally have antibodies against what is naturally on the surface of the red blood cells. So the amazing thing about having AB blood is you can give that person almost any kind of blood because they don't have antibodies floating around in their plasma that's going to cause problems. You can put this in as long as you take out the anti-A antibodies, because you can strain them out. As long as you take out these antibodies, they'll have no trouble with these kinds of red blood cells. No big deal. And I've heard that you can still put donor blood into a person's body with the antibodies for this person who has AB, as long as you slowly put it in. I guess the antibodies don't cause as much of trouble. But screening them out or taking them out is going to give them what they need. They need these red blood cells. Best case scenario, they're going to give a person with AB blood, but that is a rare blood type. O blood, let's check it out. O red blood cells. I'm just drawing it like an O because they don't have the A antigen on the surface. They don't have the B antigen, but they have both kinds of antibodies naturally floating around, their body. Both antibodies. That's why somebody like myself with O blood, I need O blood. If you give me, even if you take out the antibodies from the donor, if you give me this cell, I got these things floating around my plasma that are going to freak out and bind to it and cause that clumping. That's going to cut off blood flow to some important vital organs, and it could kill me. You can give O blood to O blood. The good news is that O blood is very common. Depending on what country you're in, the percentage of people with O blood can be a little less, but at least in the United States, O blood is the most common. Next up, Rh factor. This is either Rh positive or Rh negative, and this was named after the fact that it was discovered in the rhesus monkey. The word rhesus, not like the candy, is Rh, R-H-E-S-U-S. So in the rhesus monkey, they discovered this. This comes from a separate gene. Whether or not you're A, B, O, or AB, that comes from a certain genotype you inherit. This is on a different gene. And think of it this way. If you are Rh positive, you have an additional antigen on the surface. By the way, before I move on and forget, the O red blood cell, I don't want you to think it has no antigens at all. There are lots of different kinds of antigens. And the surface of even my red blood cells are going to have antigens projecting out from the plasma membrane. They're just not going to have the A and B antigens. But back to Rh factor, if you're Rh positive, you have an additional antigen. Think of it as like this. Let's say this is an O red blood cell. Here's another O red blood cell. If you are Rh positive, you might have this additional little antigen sticking out from the surface. If you're Rh negative, you don't have any of them. You don't have that particular antigen. It's the lack of the antigen. That's why it's negative. The way this comes into play is, I told you earlier that AB blood, if you have that, you can be given almost any blood type. The reason why it can't be any oftentimes is if you are AB negative, that means your AB cells, your AB red blood cells, don't have this particular antigen. And if you introduce, let's say, O positive blood, your body could actually end up attacking those red blood cells with antibodies because you're not used to it. So if you're AB negative, they need to give you O negative or B negative or A negative. I'm O positive, so they can give me O negative or O positive. Somebody who's O negative, they don't give O positive to that person. You know, if you're in an ER and they need to give you a blood transfusion, within a matter of minutes with a test, they can figure out which of these blood types you are. Sometimes they don't have minutes. Sometimes you have somebody who's bleeding profusely, close to death. In that case, they will give them O negative blood if it's on supply because they don't have time to wait. So they're just going to give them that one. But if there is time, if they know a surgery is coming up, they'll try to get the precise type of blood that matches the blood that's naturally in their body. Here's a little chart that you can study that tells you which one goes with which in terms of the donor giving it to the recipient. Like I said earlier, if the donor is O positive, like myself, I cannot give to O negative. But guess what? O negative can give to me. So if I'm the recipient, I can actually get O negative or O positive blood. The yellow sections have to do with, if it's light yellow, they're saying that you can give them plasma transfusions, but not with the whole blood. Bright yellow, that's full blood transfusions. White means no plasma compatibility, meaning they would give that person what's called whole blood or just the hematocrit. Hematocrit's actually a better name for it. That's the compacted red blood cells. So there you go. White blood cells, also known as leukocytes. Leukocytes come in many varieties and have an incredible ability to defend the body. Here are the main functions, or abilities rather. They can migrate out of the bloodstream. Amazingly, a white blood cell can squeeze itself. If you go inside of a capillary, they can squeeze themselves through those endothelial cells into organs. And that's amazing, their ability to migrate and hunt down these foreign bodies that could cause problems. They have amoeboid movement. If you've taken biology, amoebas, they have this ability to manipulate the shape of their plasma membrane and make what look like are called pseudopodia, look like kind of fake feet sort of extending out. And that's by manipulating the cytoskeleton. When they move their microfilaments, that part of the cytoskeleton, those protein strands, they pull and let go on parts of the plasma membrane, and that allows them to wrap around and swallow particular foreign invaders. It also aids in their ability to squeeze out of blood vessels by narrowing themselves. They're attracted to specific stimuli. Depending on what white blood cells you're talking about, they're able to hone in on certain kinds of foreign bodies, bacteria, certain viruses, even parasites. And most of them do phagocytosis, which means cell eating. It's a form of endocytosis where they are wrapping their plasma membrane around that foreign body, taking it in, and then that little sac, which now contains the bacteria or what have you, will then fuse with lysosomes, little cellular organelles that have enzymes that can digest that particular foreign invader and kill it. So there's some abilities of white blood cells. The first class of white blood cells I'd like to tell you about is granulocytes. And like I said before, they have little granules in their cytoplasm. And a lot of these granules you're seeing is lysosomes and enzymes. A lot of them are meant to be there because these are cells that kill. Neutrophils, about 50 to 70% of white blood cells, very common. They're meant to swallow up foreign invaders, and they're typically on the front lines. If you have an infection, they're usually the first ones there, and considering how many of them there are relative to the other white blood cells, the chances of them getting there first is very good. So they are there on the front lines, like they use that term with a war, with an army. Eosinophils, a lot less, 2 to 4% of white blood cells. They attack objects with antibodies. So if antibodies, these globular proteins, have been attached to a certain bacterium, eosinophils notice it, it's got antibodies on it as like a marker, and they go and eat it up. They're great at attacking parasites. If you have a parasitic infection, like let's say a tapeworm, your eosinophil count is going to go up. Because they're good at attacking parasites. And they increase in number during allergic reactions. And allergic reactions really just depends on the person. Allergic reactions probably happen because of a combination of genetics and environment affecting that abnormal immune response, but eosinophils do play a role in that. And you can see that each of these pictures, they have this weird thing going on in the middle. It's called a multi-lobed nucleus. It's like a nucleus with a few different sections. Neutrophils oftentimes will have many lobes. Eosinophils classically have a bilobed or two-lobed nucleus. And then basophils, the smallest percentage of granulocytes, they assist with mast cells in producing inflammation, expanding blood vessels in an area, and that is a classic response when you have an infection or damage to an area. Agranulocytes is the opposite, meaning they don't have those granules. They look not quite as like they don't have as many dots in them. Monocytes is one type of agranulocyte. That's about 2 to 8% of white blood cells, just depending. They are the largest white blood cells. They are large compared to the others. Great at endocytosis, like I mentioned before, with kind of the amoeba action wrapping around and engulfing bacteria and what have you. They tend to circulate in the bloodstream for about a day, and then they end up in a tissue outside of the bloodstream as a macrophage. A macrophage means giant eater. Like I said, they are quite large, and macrophages, you'll find those roaming around in tissues, eating up stuff that shouldn't be there. Lymphocytes. About 20 to 30% of white blood cells are lymphocytes. They circulate in the blood, of course, but they also hang out in lymphatic organs like the spleen. The thymus is another one that has to do with it, but the spleen is going to be a place that you're going to find a lot of lymphocytes. Here are some examples of lymphocytes. T cells have to do with cell-mediated immunity. So T cells, they are going to be targeting particular invaders as they come in. T cells, some of them are called helper T cells. You may have heard that term before because HIV is a virus that has the most impact on the helper T cells. Now, HIV apparently can invade a lot of different kinds of white blood cells, but the clinical problems associated with HIV and AIDS has mostly to do with their impact on helper T cells. There's a certain kind of protein on the surface of those cells, and HIV basically has the key that fits into the lock that allows it to get into those cells and cause problems. B cells are a little bit different. They have to do with the manufacturing of antibodies based on things that you're exposed to, based on certain viruses, certain bacteria that you're exposed to. And I remember that because antibody with a B, B cell antibody. And lastly, natural killer cells. They're natural killer cells because they roam around the body and they target abnormal cells in your tissues. So if you have some odd cell growth that's not correct, it's not healthy, they will eat that tissue. Having good natural killer cells makes it less likely you're going to get cancer. And I mean getting long-term metastasis, cancer that grows and spreads, because if you have deficiency of your natural killer cells, you're going to get out of control cell growth eventually, and natural killer cells help with that. Platelets. Thrombocytes is the technical term for platelets, and they do look like pieces of a shattered plate. It's like you took a plate and then all these little pieces, all these little red parts clumped together, those are platelets aggregating, coming together. These cells do have many important roles related to clotting blood. They release chemicals to help encourage the occurrence of a clot, of that plug in a sense. They form a temporary patch on the walls of damaged vessels. So if you have a cut that is causing the leaking out of blood, platelets are what initially come there and kind of patch it up. And they help in contraction to reduce clot size. So eventually, you want to eventually start to make the clot not as big, and so they help get the clot kind of flatter and reducing its size once some healing has begun to occur. Hemostasis basically means like keeping blood where it should be. It's the stoppage of bleeding, of hemorrhaging. There are a few different phases. When you have a cut in a blood vessel, the first thing that's going to happen, they call it the vascular phase. There'll be certain stimulation that causes that blood vessel to decrease in diameter, at least for a bit, because the immediate thing you want to do is stop a lot of blood from getting out of there, so reducing the width of it is going to do that. And it's endothelial cells right next to where the blood is. Remember, if you saw the blood vessel lesson, endothelium is where all the blood is rushing through, that inner layer. They're going to release those chemical factors that have to do with that. Next is the platelet phase. You have platelets in your bloodstream just flowing all around, and then they are stimulated to aggregate there, to come there and kind of bind to one another. And it's almost like glue in a sense that's trying to get that vessel patched, to get it so that you don't have as much blood coming out of it. And that's going to cause the release from the platelets of more chemicals like ADP and clotting factors. ADP, if you've taken biology, this is the same ADP that you get when ATP, adenosine triphosphate, is broken apart. This is adenosine diphosphate. And that is a chemical factor that can actually encourage the formation of a clot in your bloodstream. Next is the coagulation or blood clotting phase. So the platelets have started to patch up this vessel cut. But you also want to do a better job of patching up that area so that you don't have bits of blood leaking out through it. So in addition to platelets, fibrinogen, one of those globular proteins, is converted to fibrin, and it forms a net-like structure that kind of catches red blood cells, almost like fish being caught in a net. And as they're caught in this net, it forms a clot, a plug that helps stop blood from exiting. Now, eventually, once that blood vessel wall is completely healed and you have new connective tissue, making it nice and sound, eventually you want to get rid of the clot. You don't want the clot to grow and hang around too long and reduce blood flow through that vessel once the healing has happened. So fibrinolysis, if you want to call it that, means getting rid of that fibrin net, getting rid of that clot, and making it go back into its little bits so that you don't have that big clump there when it's not needed anymore. Finally, some blood conditions and disorders. Some of this overlaps with the blood vessel lessons from before. I actually am using the same pictures here because blood is running through blood vessels. Hemorrhaging is the leaking of blood out of blood vessels. Internal hemorrhaging can be a very bad thing, obviously. Thrombus is a blood clot that shouldn't be there, and if it gets dislodged and eventually moves along through the bloodstream, that's an embolism. And an embolus gets caught and becomes an embolism. Depending on where the embolism happens, that can restrict blood flow to the lungs, to the heart, to the brain, and it could result in a fatality. Anemia basically means that you're not producing enough red blood cells, or the red blood cells you are producing aren't getting the job done. So the main way people get anemia is they're not taking in enough iron. You need iron to produce hemoglobin, which is a critical part of red blood cells. So if your iron intake is low, you're going to feel fatigued and tired. And there's other long-term health effects that can happen because of anemia. And being chronically anemic can shorten your lifespan. Doctors will tell you if you're anemic, eat more red meat. And that's a problem for a vegetarian, obviously, but red meat has a high iron content compared to some other meats, and that can encourage your body to make more red blood cells through erythropoiesis. Sickle cell disease is also called sickle cell anemia. This is an example of where you may be producing the right amount of red blood cells, but the red blood cells you're producing aren't doing a good job of binding oxygen, and they may be getting caught where they shouldn't. So sickle cell disease or sickle cell anemia is something you're born with, and there is a gene corresponding to the shape of hemoglobin. And the amazing thing is that all the amino acids that are put together to make hemoglobin, it's a point mutation. It's one DNA base that's wrong, and that impacts one of the amino acids and changes the whole structure of hemoglobin, so that instead of the cells looking like this, that round shape with that little indent, that little well, they kind of look like this, like a sickle. And that shape means hemoglobin isn't doing as good of a job at binding oxygen effectively, and this shape also causes these red blood cells to get caught in certain parts of the body. And there are long-term negative health effects of having sickle cell disease or sickle cell anemia. Hemophilia is another genetic disorder. Unlike sickle cell disease, which is an autosomal disease, meaning not sex-linked, hemophilia, the most common form, is sex-linked. It's on the X chromosome, and if you've taken biology, you might remember that because it's on the X chromosome, it affects more males than women. Males only have one X chromosome, females have two. So all it takes is one harmful recessive allele on that one X chromosome in men to give them hemophilia. It takes the inheritance of that allele from both parents in women. So hemophilia, this literally means loving blood. And ironically, someone who's hemophiliac would not love to see blood. The reason why it has that name is because when they get a cut, blood keeps coming. They do not have the ability to properly, naturally clot their blood. That's a problem. There are so many different factors. There's something like 20 factors that have to do with this domino effect that leads to the formation of a clot. I gave you a very brief summary of how a clot is formed, but it's a very complicated chemical process. And so being a hemophiliac, something is wrong with that sequence of how a clot is formed, and you don't have the proper blood proteins to do that. There is no cure, but if you are a hemophiliac, you can carry around something that allows you to inject clotting factors when you do get a cut, or you're going to have to take a trip to the ER to deal with that. Leukemia is a bone marrow cancer, and leukemia, one of the initial signs of it is actually overproduction of leukocytes, because what all cancers have in common is it's mitosis out of control. It's too much growth within that tissue, and that can negatively impact neighboring healthy tissues. So cancer of the bone marrow, you're going to end up producing way more white blood cells than you should. And leukemia is fatal if that person doesn't get a bone marrow transplant. Thanks for watching this blood lesson, and thanks for watching educator.com.